This biochemical method allows for the determination of the palmitoylation state of any membrane protein expressed in the brain for which a suitable antibody is available. The main advantage of this technique is that it does not require affinity purification and instead utilizes changes in gel mobility to determine the number of modifications of a protein of interest.
After dissecting out the brain, homogenize it immediately in 10 milliliters of homogenization buffer in a glass homogenizer on ice, using approximately 12 strokes. Centrifuge the lysates for 15 minutes at 1400 times G and 4 degrees Celsius. Transfer the supernatant to a new tube on ice. Resuspend the pellet in 10 milliliters of homogenization buffer. Homogenize it using approximately 6 strokes. Centrifuge the suspension at 710 times G and 4 degrees Celsius for 10 minutes. Combine the supernatants and centrifuge them at 40,000 times G and 4 degrees Celsius for 20 minutes. Discard the supernatant and resuspend the pelleted membrane fraction in homogenization buffer by breaking up the pellet with a pipette tip and pipetting up and down. Perform a BCA assay to quantitate protein levels. To perform the APEGS assay,